Good morning. Well, back here for Chapter 9, Genetics of Bacteria and Viruses. So, why do we care so much about these guys? From a science point of view, viruses and bacteria are really amazing research tools, actually. You can put new genes into viruses and bacteria and study how those genes work. From my point of view, that's kind of, how I've used them as a tool in research and in studies in lab work that I've done as a graduate student and as a postdoctoral fellow. But in addition to that, there's also the health aspect of bacteria and viruses. So we know that bacteria and viruses can make us sick. Bacteria in and of themselves can be a major reason for people developing infections. There are also viral infections, though. A bacterial infection can be kind of handled using antibiotics, and we'll talk about why that's the case. Viral infections, those are a little bit different. Viruses, you cannot kill using an antibiotic, and we'll talk about that a little bit too. Now, viruses and bacteria are not, they do not fall under the umbrella of eukaryotic organisms. Bacteria are prokaryotic, as you remember. So I think of that as bacteria are prokaryotic or professionals at making you sick. You are a eukaryote. So the bacteria, the professionals, infect the eukaryotes and basically make their home inside of a living eukaryotic system. So when somebody has a sinus infection, you have a bacterial infection up here in your sinuses and down here in your nasal sinuses, in the sinuses above your eyes. So, bacteria, it's worth understanding the genetics of bacteria and viruses simply for their impact on health. And so it turns out that many of the genes that allow bacteria to survive in your sinuses or in your lungs or under your skin, many of those genes have different alleles. And so it's the allelic differences that allow organisms to have antibiotic sensitivity or antibiotic resistant. And I'm sure that you guys have all heard about different antibiotic resistant organisms. This is really important because understanding how antibiotic resistance develops in bacteria will help you understand how to stay well. So let's start with the bacteria first. Bacteria are haploid. In other words, they don't have a mom and a dad. Remember, bacteria divide through binary fission. They divide in half. So they only have one N, okay? So remember, haploid is 1N. Diploid is 2N. So when you have a bacterial cell, so here's my idea of a bacillus-shaped bacterial cell. It's real simple. They have their own individual chromosome that is shaped like a donut, a giant donut. And it's so large that it actually doesn't look so much like this round circle kind of, loopy circle I made here, but it looks more like spaghetti inside the cell. All right. And it may or may not actually be attached to the interior of the cell. Don't worry about that right now. Now, it turns out that there are lots of bacteria strains that have what we call loss of function mutation. Loss of function mutations are mutations in a gene that render the gene useless. So if an organism gets a recessive mutation, that might be the mutation, a loss of function mutation that knocks the function of that gene out. We'll get to that more in a little bit. Now, remember, bacteria reproduce asexually, which is the same thing as binary fission. Okay? The binary fission that we talked about earlier on in the mitosis-meiosis chapter. There is no crossing over because there is no meiosis. Remember, no fertilization to make a bacteria. So how do you get genetic transfer in an organism that doesn't make gametes? Well, bacteria have a few cute little tricks that they use to exchange information with other bacteria and exchange genetic traits. So it turns out that you can get a piece of bacterial DNA and transfer it from one bacteria to another, but it's not through sexual reproduction. It's through one of the subjects that is in your discussion this week. So please go look at that discussion and identify one of the three mechanisms for gene transfer in bacteria. We're going to cover that in today's lecture. So, like sexual reproducing eukaryotes, genetic transfer in bacteria increases the genetic variability or diversity in the pool of bacteria that you're dealing with. And when I say a pool, sometimes it can literally be in a pool, or sometimes it'll be inside of tissue, let's say under your skin. So, genetic transfer in bacteria happens three ways: conjugation, transduction, or transformation. Conjugation, just so that we are clear right now, this is not bacterial sex. Okay, it is not bacterial sex. Some people will say that kind of as, to make it easy for you guys, or to make it kind of more understandable. Conjugation is not when fertilization occurs, okay? Sexual reproduction involves fertilization. This is involving the transfer of DNA from one cell to another, but by the cells physically touching each other, and we'll get to that. Transduction is where viruses transfer DNA from one cell to another. So basically, you're going to have one cell here, one bacterial cell, and one bacterial cell here. And let me get a different color here for my virus. It doesn't want to give me another color. Where'd my colors go? Doggone it. Well, here's my virus. Okay, and this would be a bacterial phage. The virus will infect this cell and then take DNA to this cell. We'll talk about how that happens. And then transformation is really interesting. This is where bacteria literally absorb DNA from the environment. And that's kind of a scary thing, because that implies that bacterial cells have the ability to take naked DNA up from like the counter, absorb it, and then use it. And that's actually what happens. There was a research article produced at UC Merced where the professor, the researcher, was looking at what types of bacteria are growing on the walls in the local hospitals. And it turns out that the bacteria were undergoing transformation and conjugation on the walls of the hospital, which means that if you have one bacterial cell here that is antibiotic resistant and one that is not, when they get together, this piece of DNA that confers antibiotic resistance will transfer from one cell to the other, making this cell antibiotic resistant. Sorry, I'm still trying to figure out the whole camera angle thing here. So this is a really nice figure, actually, identifying conjugation, transduction, and transformation. So table 9.1 and slide 5 are really good to study together so that you'll get an idea of what these processes are. Let's start with conjugation. So conjugation started back in the early, understanding what conjugation is started in the early 1940s. Edward Tatum in his lab was trying to understand how bacteria change, how they acquire different traits. And Joshua Lieberg was also working on this. Now, Edward Tatum is later on, as you'll see, was working with another professor whose last name was Beadle. And we'll talk about the very famous Beadle and Tatum experiments. But basically what what Liebenberg and Tatum were working on were looking at strains of E. coli, okay, and looking at what we call nutritional growth requirements. In other words, every organism has to have a specific set of amino acids. Now remember, amino acids make proteins. Okay, I hope this thin pen is working okay. You have to have numerous amino acids held together by peptide bonds to produce a protein. So each one of these little circles is an amino acid, and the whole thing is going to be a protein. Each one of these lines is what we call a peptide bond. The peptide bonds holding the amino acids together. So it turns out to make proteins, E. coli, just like humans, need to have all the amino acids required to make protein. Well, E. coli don't eat the way we do. E. coli do absorb amino acids from their environment, and they can convert one amino acid to another. So they can make like leucine into threonine. Actually, it's threonine to leucine. But Some of these organisms, some E. coli, aren't able to make that transition. So if you cannot convert, let's say, proline to isoleucine, then that organism is called an autotroph. In other words, the organism is unable to take one amino acid and convert it to another. When that happens, then that particular cell has to rely on the environment for absorbing isoleucine. And if it's not in the environment, then the organism will die because it cannot make proteins that have isoleucine in it. So, what are we talking about here? Number one, the organism has to have all the nutrients in the environment to make a protein. Those nutrients are amino acids. Number two, of the amino acids that the organism absorbs, it has to be able to convert some of those to different amino acids, okay? Because sometimes you're not going to find those different ones in the environment. If the cell cannot convert amino acid 1 to amino acid 2, then it's going to lack amino acid number 2, and it then might die because it can't make the new proteins that have amino acid number 2 in it. So that's this example of proline to isoleucine that I put here. Autotrophs cannot synthesize all the nutrients that they need. Therefore, all those nutrients have to be in the environment. Now, you have the other group that are the prototrophs. Prototrophs have the ability to make all the nutrients they need from nutrients they absorb in the environment. So basically, they can convert proline to isoleucine, methionine to, actually, it would be like, I don't know, valine to methionine, let's say. So, auxotrophs need help. They need supplementation. Prototrophs can make whatever they need from substrates in the environment. So, in this particular experiment where Tatum and Liebenberg were working, they had one auxotrophic strain of E. coli and another auxotrophic strain. Now, what the first strain was able to do was it could not make biotin. Let me erase this here. Okay, so we would call that BIO negative. It could not make methionine. It was methionine negative, but it could make threonine, it could make leucine, and it could make thiamine. Okay, so the cell can make these. And I do A dot for amino acid. So it could make these, can't make these. Wow, I gotta say, my handwriting looks terrible. Over here, so this is strain number 1. This is strain number 2. It can make biotin, so it's bio plus, it's methionine plus, okay? It can make these. Let's separate these two here. Over here, it's threonine negative, leucine negative, thiamine, THI. Sorry, negative. Cannot make these. Okay. When we look at how you designate this when you're writing it out, it's easier to, like I kind of wrote it out for you, bio negative, methionine negative, and so forth. In this experiment, what they wanted to know was, could strain number 1 pass information to strain number 2, okay, and vice versa? In other words, can this group, these genes, be sent to this cell type, and could these genes be sent to this cell type? And so they were looking at whether or not the strains could pass these traits to one another. And what they did was they made agar plates that were selective. So some plates would have threonine, leucine, thiamine, or would be lacking other things. So you'd have number 1, plate number 1, and plate number 2. Don't get that confused with this group here. All right. I'm going to stop here so I can start the next lecture and go through how the experiment was actually done. For quite a few lectures now, we're going to be talking about real experiments. These were experiments done in real labs. These are seminal experiments in genetics and in microbiology, and it's really important to understand, number 1, how the experiment was done, number 2, how it works, and number 3, what the result means.